I think, understanding how information is stored in the brain. 45 years ago, we started out by finding how is information stored in genetic information stored. So we now want to know how information which reaches us by various forms of perception is stored and retrieved. So that's an enormous challenge, and in part we don't understand, how the brain develops.
In the sky, we discover stars by their light and by their light alone. Stars so distant from each other that no material thing could ever have passed from one to another. And yet, this light, which is to us the sole evidence of the existence of these distant worlds, also tells us that each of them made up of molecules of the same kind as those which we find here on Earth. A molecule is a group of atoms. If the atoms are not all the same, a molecule is called a chemical compound. Some are simple like water, many are complex like most organic compounds.
Our world is full of machines, non-living entities assembled by human beings from components humankind has made. Our automobiles, computers, furniture, and even screwdrivers far too outnumber us. Despite this proliferation, no machine can reproduce itself without human agency, at least not yet. It is predicted that in the 21st century, scientists will introduce a manufacturing strategy based on machines and materials that will virtually make themselves. This process is called self-assembly. Let us understand what is self-assembly. This approach is based on the recognition that every object is a collection of atoms and molecules arranged in an ordered fashion. Self-assembly tries to get atoms and molecules in an aggregated order to do this without human intervention. Look at this water drop. It has a smooth curved shape of just the kind required for optical lenses. Grinding lens of that kind would be a major undertaking, yet the liquid assumes that shape spontaneously because the molecules at the interface between the liquid and air are less stable than those in the interior. The laws of thermodynamics require that the raindrop takes the form that maximizes its energetic stability. The smooth curved shape does so by minimizing the area of the unstable surface. The idea of designing materials with a built-in set of instructions that will enable to mimic the complexity of life is highly attractive. Researchers are now beginning to understand the kind of structures and tasks that could exploit this approach. Coded self-assembly is truly the concept for the next century. Does it sound like a science fiction? You would be surprised to see a sequence from a science fiction film made 3 decades back, where human beings are transformed to the size of molecules.
19 seconds, Doctor.
The semi-lunar valve.
Hey, see it, Captain? Yeah. I see it now.
This imagination and dream is not far from reality, as science succeeds in developing smaller and smaller nano devices. You know, scientists are very disappointed that computers are not more intelligent. This is where the new discipline, biomolecular electronics, comes in.
Liquid crystal displays have become the display of this age because they are the lowest power consuming displays known. They consume only 1 microwatt of power per centimeter squared of displayed area. Liquid crystals consist of small, organic, anisotropic molecules. In fact, in the future, all computers are said to have only liquid crystal displays because they are much better for the viewer. It doesn't produce any X-rays, for example, and therefore it is considered to be safer to use than the usual CRT displays that we are now using. So in the new information age, liquid crystal displays are certain to play a very leading role. And since these are made of simple organic molecules, this is the first example of molecular materials which have made a big impact on the electronics industry.
I feel that molecular electronics is one of the most important areas today because this area has added a new dimension to our effort in producing new materials and new devices. For example, in nonlinear optical materials, for also many other areas of materials where instead of a conventional inorganic, ceramic, metallic materials, we can use molecular materials. For example, one of the most promising things that has come out of it is using an organic material for producing laser type beams, blue light, for example.
Of lately, there has been a very big discovery and invention in which you can see light from a polymer, plastics, which are normally not used for such crazy applications. But of lately, it has been seen that these can act as semiconductors and can give you any color of light you want with very good efficiency and with great packaging feasibility options. Now, this ability of the polymer to both emit light and absorb light and convert it into current is this is one of the routes for designing alternative circuits, molecular circuits, and a pathway for optical computing.
We are even looking at metal particles. We put organic molecules around them and see how these metal particles form new kinds of lattices. They organize themselves because of this organic environment around that. That is another area. We are also designing other types of organic structures based on what is known as supramolecular chemistry. Ordinary chemistry is molecular chemistry. Supramolecular chemistry is one where molecules interact with each other by weak bonds, not usual bonds.
By the end of 20th century, science had reached the end of an era, unlocking the secrets of the atom, unraveling the molecules of life, and creating the electronic computer. These three fundamental discoveries, triggered by the quantum revolution, the DNA revolution, and the computer revolution, helped in solving the mysteries of life.
We are on the threshold of a new millennium. Human knowledge is doubling every 10 years. Computer power is doubling every 18 months. Internet is doubling every year. The number of DNA sequences we can analyze is doubling every 2 years. Almost daily, headlines herald the advances in telecommunication, computers, biotechnology, and space explorations.
India itself is putting a lot of thrust in this area, both in the general area of molecular electronics and in the area of biomolecular electronics. This being a multidisciplinary area, funding can come from agencies such as Department of Science and Technology, Department of Biotechnology, Department of Energy, and many other such areas. And therefore this field, which is still in its primacy in India, is being looked at as a futuristic area which may bring rich dividends to the country.
Biomolecular electronics will be soon on its way to becoming big business. It will make an even bigger difference not just to the world of research, but to the planet's economy and politics. Historically, many biologists are influenced by the theory of vitalism, a mysterious life force, a substance-animated living thing. This view was challenged by Schrödinger in his 1944 book, What is Life? He dared to claim that the life could be explained by genetic code written on the molecules within the cell. It was a bold idea that the secrets of life could be explained by using quantum theory. James Watson and Francis Crick, inspired by Schrödinger's book, eventually proved his conjecture by X-ray crystallography.
It really began to take off, I think, when we, with the concept of messenger RNA. And that led to the code. And of course, the big step forward was recombinant DNA, the ability to move DNA from one organism to another, to take a DNA molecule, cut it in pieces, rearrange those pieces with DNA from another source. That's really what is, that's behind the growth of biotechnology. Without recombinant DNA, we'd have a series of rules understanding, but we wouldn't be understanding individual genes, and the physicists would still think they have the most exciting science. But everything that, there'd be no human genetics without recombinant DNA. We wouldn't know how to tackle genetic diseases.
A number of Indian scientists working in various research institutions are now working in the field of physics, biology, and electronics. Let us have the glimpse of few of them.
The most exciting thing about the biomolecular based sensors, gas sensors, is that you can integrate specificity by choosing proper biomolecule, so that only one particular kind of gas will be sensed at a time, and also quantitation is easily possible. Moreover, if you are putting more than one biomolecule, which is selective of different kinds of gases, then in that case you can simultaneously detect different kind of gases.
What we're trying to understand here is the manner in which a nerve cell is able to transmit electrical signals. And the way this is done is by permitting sodium or potassium or calcium ions to go in and out of cells. So what we are trying to do is to take these molecules, for which we have the genes cloned, and try to understand what these molecules look like. And once we have some understanding of their form, we try to understand how it is that this form is able to permit the function or to facilitate the function of these molecules. So for that, we do manipulations of the gene, put them back into cells, look at their expression, and the manner we look at the expression is by sticking in electrodes and then recording the currents that we see from them. So for instance, we can see in the recordings that are displayed on the screen there that at certain potentials there is no conductance, at different potentials there is currents that come up, they increase, they decrease, and we are trying to understand how it is that this process occurs.
The molecules that we are working on, they walk on microtubule cytoskeleton inside the cell, and they carry different cargoes on their tail, which is called the cargo just because it's like acting like any other motor or any other train on its track. In principle, it should be possible to measure the force and displacement available from these kind of molecular motors, and then they will fabricate some nanomotors which will be mimicking similar sort of force and which can be converted into mechanical energy and ultimately movement.
If you take a human being, there are close to 3 billion base pairs of DNA present. It's important that how these base pairs are arranged in the stream of DNA, that is the one which decides what function it should code for, what kind of proteins it's going to make. Now, one of the most interesting problems is the problem of what you call the control of this gene expression. You take a human being, for instance. If you take the DNA, the genes, from his cells of the brain, from the cells of his liver, the cells from his legs or from his toes, we have the same chromosomes and the same DNA present. That is, the total genetic information present in all these cells are the same. Yet, we know a liver cell is a liver cell is a liver cell, and a lung cell or a brain cell is doing a different function. The brain cell does the function associated with memory, speech, and what have you. You take the liver cell, it makes all the digestive enzymes. You take the cells of your pancreas, it makes an important hormone like insulin. So the question is not that what is the total information content. From within this total information, which is coded in the form of DNA, how specific sets of information are picked up and expressed in a particular tissue or a cell. In fact, this is what we call the differential gene expression.
During the past decade, scientists have shown increasing interest in using biological macromolecules, such as proteins, as active components in molecular electronic architecture. Biomolecules provide several advantages because nature itself has solved many problems involved in carrying out logic switching or energy transduction simply by trial and error. Among many biological materials which are found to be suitable for this kind of work, bacteriorhodopsin is found to be the most suitable. We are generally familiar with 4 biological macromolecules: proteins, nucleic acids, carbohydrates, and fats. Bacteriorhodopsin is a protein. It is called bacteriorhodopsin because it is similar to a protein called rhodopsin, and rhodopsin is the protein which is involved in our vision. It is this protein which allows us to see things around us. Simply that this protein is of bacterial origin, it is called bacteriorhodopsin.
There are other possibilities. They can be processing information optically. They can be processing information by electric and magnetic techniques. All these would be done with the size of the devices much smaller than what is the current day practice in integrated circuits, the so-called silicon or gallium arsenide IC chips. Typically, they operate at dimensions which are about 1/1,000,000th of a meter in size, 1 micron in size. But here we are talking of devices which are 1 nanometer, typically 1,000 times smaller than the IC chips that today's technology is providing. It is a long challenge. We are only beginning to understand how these devices work. And yet, not only all over the globe, but even in India, people are beginning to understand how we can start using these devices not only for simple operations that we are doing today, but hopefully for more complicated operations in the future. That is where, by joining the race right at the very beginning, we hope to be able to be competing in the international scene. In the case of silicon, gallium arsenide and other technologies, for various reasons, we were 20-25 years behind the developed countries, especially United States, Europe and Japan. But in this area, we are roughly contemporary, maybe a little late, but we have a hope of catching up with the best in the world.
There are several advantages of using biomolecules for biomolecular electronics. In case of computers, for example, since biomolecules can be designed one atom at a time, it gives engineers full control for manufacturing suitable gates. For example, while conventional two-dimensional memory on a CD-ROM can store 100 to 200 megabytes per square centimeter, a protein-based three-dimensional memory can store 100 to 200 gigabytes per cubic centimeter. If these 3D memories are used as RAM, it is felt that the protein-based computers can easily be operated 1,000 times faster than any man-made computer. There is one more issue. In nature, even a child or even a small virus actually assembles itself. It basically makes itself. This process is called self-assembly. Sooner or later we are going to want computers to make themselves, even small equipment to make itself. This kind of self-assembly requires information encoding and what is called template on which whole thing can be assembled. Both these elements are currently only possible with biomolecular electronics.
Someday soon, perhaps, scientists of all these persuasions will synthesize chemical life with computer life to produce a living being that can evolve. Will this be the ultimate reply we humans give to the silence of the universe?